Welcome back to the 10th Breastfeeding Congress with the theme, Roadmap to Breastfeeding Success, Creating Systems for Lactation Support. We would like to welcome our moderator for the simultaneous sessions on evidence updates, Dr. Maria Isabel Quilendrino. Dr. Quilendrino took her Doctor of Medicine at the UP College of Medicine and had her residency in pediatrics and fellowship in developmental and behavioral pediatrics at the Philippine General Hospital. She is currently a clinical associate professor at UP College of Medicine and is a member and technical reviewer of the research committee of the Philippine General Hospital. Again, we welcome Dr. Quilendrino.
Good day. I am Dr. Maria Isabel O. Quilendrino, and I will be your moderator for this series of lectures on evidence updates. Our first speaker is Dr. Irma Macalinaw. Dr. Macalinaw is a graduate of the UP College of Medicine. She had her residency in pediatrics at the Philippine General Hospital and underwent further training in toxicology at the UP National Poison Control and Information Service and the Rocky Mountain Poison and Drug Center at Colorado, USA. She is currently a professor of the Department of Pharmacology and Toxicology of UP College of Medicine and is a training advisor of the UP National Poison Management and Control Center. She is the UPCM coordinator for chemical, biological, radiological, and nuclear health security initiatives, and is a fellow, founding secretary, and incumbent president of the Philippine Society of Clinical and Occupational Toxicology Incorporated, as well as a board member of the Pacific Basin Consortium for Environmental Health. To talk to us about microplastics in human milk, a cause for concern, let us all welcome our first speaker, Dr. Irma Macalinaw.
A pleasant day to all the participants of this virtual workshop. My name is Irma R. Macalinaw, and I'm about to discuss microplastics in human milk. Is it a cause for concern? Our learning objectives cover the following. 1, discuss what microplastics are, its behavior and characteristics, describe the potential adverse health effects caused by the internalization and accumulation of microplastics, and lastly, to elucidate the impacts of microplastics on human breast milk practices and breastfeeding. Plastic pollution is one of the century's major issues for humankind. We live in an era in which plastic lingers in the world's ecosystems and infiltrate the human food chain and eventually enter the human body. But what are microplastics? You can see that environmental plastic pollution, depending upon the size, may result in what people call mesoplastics, microplastics, and of course, the last one, nanoplastics, which is actually one of the most difficult to determine as yet because of the lack of instrumentation. And therefore, what we can see and measure at the moment really are microplastics in the range of 1 micrometer to 5 millimeters. So microplastics are now considered to be a major environmental threat since they are prevalent in practically all ecosystems. To understand the effects of microplastics to human health, it's also important to be able to identify the different forms according to its source. For example, when we talk about microbeads, these are tiny polyethylene beads which we commonly find in exfoliants in health and cosmetic products. They can be generated also from industrial cleaning agents such as those of scrubbers. Plastic powders generated from powders used in industrial moldings can also produce microplastics. Our fibers come from clothing and cigarette filters. And of course, you also produce microplastics from the styrofoam cups and also in the wear and tear of tires. But there can also be degradation of bulk plastic, and therefore this can result in what people call as the secondary microplastics. So what are the characteristics of the microplastics found in human biological samples? In this slide, you can see that it depends upon the type of microplastics. You can have different shapes, colors, and of course, what you find in the human biological samples, they range from 700 nanometers to 5 millimeters. The shape can be that of a fiber, and fragments predominate in terms of shape. The color is usually white and blue, but you can have other colors such as red, magenta, yellow, green, and the like. So the size distribution reflects the larger microplastic sizes, which are excreted mostly in dermal contact portions. And of course, when you talk about the smaller sizes, these are easily translocated within the human body. So this slide, even if it looks very busy, I just want you to focus your attention in this portion where you can see that there are different parts of the human body already affected by microplastics. You can actually find them in different parts of the human body. So our concern for today, of course, is that of breast milk. And when you talk about the most common types of microplastics that have been documented by Raman microspectroscopy, the most common one would be that of the polyethylene, the polyvinyl chloride, and polypropylene. So these particular microplastics have been found in the first report of human breast milk published in Polymers. There can be multiple exposure to microplastics, and you can see from this slide that the exposures can either be from air, water, and food. But it seems that the reports show that those which come from ingestion and inhalation are the more common routes of exposure for microplastics, with that of ingestion as the most prominent reason for why microplastics can now be found in the human body. So you can have exposures coming from ingested personal care products, such as the toothpaste and the polyethylene in the lip gloss. The dental polymers as well can be a source of microplastics, including that of the deliveries, polymeric drug delivery systems, which are actually nanoparticles. So we are exposed to microplastics now for an extended period of time. By ingestion as well as inhalation. We are exposed to over 45 different polymer types, which is concerning, and the effects are not yet fully elucidated or recognized at this time. So we need to be able to do more studies and be able to understand better how these potential hazards connected with these plastics can actually impact the human body. So this particular study that alerted many people at the time of its publication, different sectors of society, was actually done in Italy, and 34 breast milk samples were actually investigated, and the presence of for the presence of microplastics, and we can see here that 26 out of the 34 women who submitted their samples of breast milk actually were positive for microplastics. Again, as previously discussed, the shape, the dimension, color, and chemical composition was actually described by the authors. Almost all of the microplastics that were found in breast milk were blue and orange-yellow in color. They are irregular fragments with dimensions ranging from 2 micrometers to 12 micrometers, consistent with what is known as the translocation mechanism. Similar with other studies reported in literature, the most abundant polymers were polyethylene, polyvinyl chloride, and polypropylene. But it is also important to remember that there have been previous studies that showed some kind of chemical contamination of human milk, and these included the PCBs, organochlorine pesticides, the PBDEs, phthalates and phthalate metabolites, and now recently we are also seeing a lot of the per and polyfluoroalkyl substances, or the PFOS and the PFAS, which actually are the lining that are found in the boxes of food deliveries that we have been getting during the pandemic. Then we also have phenols and heavy metals. Why am I showing this particular slide? It is because even in the presence of these particular chemicals, there was no recommendation at all that says you should stop breastfeeding. But what are the potential adverse effects of microplastics in human milk? Although it is well established that humans are now exposed to microplastics, there exists a wide knowledge gap on the consequences of microplastics in humans. Following ingestion of microplastics or, again, exposure via the inhalational route, we can still see that there is a gap as to how much of this particular microplastics are really internalized and how they will affect the human body. Studies showed that microplastics, which are less than 20 micrometers, can infiltrate the organ systems, and those with less than 10 micrometers can actually cross various biological barriers and accumulate in vital organs such as the liver, the heart, and even the fetus. This slide is actually a summary slide which talks about how the microplastics can actually be degraded and through weather systems as they are dumped in sites where, like, such as landfills, and you can see here the formation of the microplastics. And eventually, these microplastic particles are actually dispersed in the atmosphere, and therefore people can actually inhale them. You can also have dermal contact, and of course, from the marine ecosystems, as the marine life actually ingest or take them up, this is now carried to the higher level of the food chain, and there can be trophic transfer, and this can also be by ingestion, and so on. If the microplastics now contaminate soil and groundwater, then you can also have exposure following ingestion and dermal contact. But what is the consequence then when it comes to health? So as I've said earlier, there is a gap in the actual knowledge as to how this would impact human health. But there are postulates that the formation of reactive oxygen species from microplastic can actually, again, result in target organ toxicity, such as neurotoxicity, pulmonary toxicity, cardiovascular toxicity, hepatotoxicity, reproductive toxicity, immunotoxicity, nephrotoxicity, and genotoxicity. So this slide is actually just a representation of the possible mechanism of reactive oxygen species-mediated cellular damage from microplastics and nanoplastics. So the entry of microplastics into the human body by ingestion, inhalation, and dermal contact can actually result in cell-mediated responses, including that of endocytosis, microcytosis, phagocytosis, and passive diffusion. After which, when these are now taken up by the cells, this is again taken up by lysosomes for further processing and released into the cytosol. It was then followed, and this is the postulate, followed by the damage of the mitochondria, increasing the reactive oxygen species burden. So enhanced reactive oxygen species results in the compromise of the antioxidant defense system and leads to protein oxidation, lipid peroxidation, and again, for the long-term effects, it causes DNA damage. So what does that mean? As I said, there is a gap, and therefore we need more studies. There's a need for human population studies. We need to conduct epidemiological investigation to assess the relationship between microplastic exposure and human health outcomes, investigate potential association between microplastics and conditions such as inflammation, oxidative stress, immune dysfunction, developmental disorders, cancer, and other chronic diseases. So the investigation on the health effects may center on the genomic effects. As we've said, there is DNA damage. We want to explore the impact of microplastic exposure in the human genome and investigate whether microplastics can really induce DNA damage, mutagenesis, or chromosomal aberrations. And of course, we want to be able to find out whether they can produce epigenomic modifications, such as those induced by DNA methylation, histone modifications, and the non-coding RNA regulation. So in the long run, we will be able to look at the long-term effects of microplastics to human health. But what can be the potential impacts of microplastics to human breast milk practices and breastfeeding? So here, this is just, again, a summary slide that talks about the detection of microplastics in the placenta, meconium. This was a pilot prospective study published in 2023. You can see here examples by which a pregnant woman can actually be exposed to microplastics. They can come from scrub cleansers, your toothpaste, and even water. And so eventually it ends up in the placenta. And when the baby is born and the meconium is investigated for the presence of microplastics, this can now be documented in the meconium of the infant. So eventually, as the infant grows and breastfeeds, there can also be the possibility of exposure from the feeding bottles. And we can see here that infants who are now breastfeeding or are being given breast milk in feeding bottles can actually be exposed again to microplastics. So the study of the mother-to-child transfer by breastfeeding was actually done in order to be able to determine whether the breast milk fed group had relatively higher abundance of microplastic compared to those with mixed feeding. So they have seen that depending on the duration of breastfeeding, the child's body burden reflects the mother's exposure as well. So the abundance of microplastic in infants who consumed breast milk greater than 600 ml per day was relatively higher as compared to those who did not. In contrast, the abundance of microplastic in infants who consumed complementary food greater than 50 grams per day was significantly lower as compared to those who did not. So, exposure to microplastics in lactating infants may occur through breast milk consumption other than the complementary food consumption. However, when we compare the microplastics between breast milk and infant milk, the authors actually did not observe significant differences between them. Therefore, there might be other reasons. Many mothers tend to use the breast pump and preserve the breast milk by breast milk storage bags, which may lead to microplastic migration into breast milk. So this slide actually shows more information in a study that was done on the release of microplastics from breast milk storage bags. And they can see that particles in the submicron range with spherical or oval shapes were present actually in the breast milk that were contained in the storage bags. Thus, infants may be exposed again to about 0.61 to 0.89 milligrams per day of microplastic particles through the use of the storage bags themselves. And again, the question would be, what would happen to the microplastics or the nanoplastics consumed? They go into the bloodstream, the lungs, the placenta, and again, the question would be, what are the health risks to the infant? So aside from the use of breast milk storage bags, there is also a study that looked at baby feeding bottles. And again, this can be a potential source of microplastic exposures with the breast milk, again, being contained in the infant formula feeding bottles. So it is actually the preparation of the polypropylene bottle feeding bottles. So there can be the abundance of microplastics in infants who have been fed with baby bottles greater than 5 times per day, and the consumption of milk by bottles of more than 500 ml per day was relatively higher as compared to those who did not. However, considering breast milk and infant formula were both prepared in feeding bottles, the authors couldn't separate them completely. Therefore, the study assumed that feeding bottle was more likely a potential exposure source for infants. So what is this particular study saying? It's actually saying that when breast milk is actually put into the feeding bottles, there is actually an additional potential source of exposure. So why is this so? They have seen that the procedure of sterilizing the feeding bottles may be a contributory factor. So further studies are actually needed when we look at the way we sterilize the feeding bottles, such as procedures such as soaking in boiling water or sterilized by alcohol, steam, UV radiation, actually increased exposure levels of microplastics in infants. So again, there was another study that used high heat. So for example, sterilization and exposure to high temperature significantly increased the release of microplastics from the feeding bottles. So in summary, I would like to be able to summarize this by saying that when we talk about microplastics, there can be different sizes, there can be different shapes, there can be different colors, a spectrum of colors. And again, the origin can be either a primary source, such as what we discussed earlier, the microbeads from cosmetics, and there can be secondary sources from bulk plastics. Again, when we talk about exposures, we know that the mother can be exposed and eventually this can be transferred from mother to the developing fetus. And during breastfeeding, we know that they are usually lipophilic as well, and therefore they can accumulate in the fatty tissues of the breast, and where the breast milk is eventually transferred to the infant during breastfeeding. But aside from this, it is also important to consider the environment of the child, because even the child himself, aside from the milk that he's taking in, can actually be again exposed from toys that may be made of plastics. It's very important to share this study published recently in the Environmental Health Perspectives for you to be able to again have a better understanding of children and microplastics. What about the way forward? I know this was not mentioned earlier as a learning objective, but I'd like to share that this is important now for pediatrics, for people in OBGYN, the greater community, research community, to work together to be able to do some kind of risk assessment and therefore integrate the findings from exposure assessments, propose mitigation strategies that will help us curb the microplastic pollution. Again, we need to be able to improve waste systems so that we do not generate so much of this microplastics, which is currently again becoming more and more present or ubiquitous in our environment. And there must be an interdisciplinary collaboration between researchers, environmental and natural sciences, genomics, toxicology people, public health, again to be able to advance knowledge in relation to microplastics in human health. So in response to the growing devastation of plastic pollution, the United Nations Environmental Assembly is advocating for a legally binding global plastic agreement by 2024. Why am I focusing our attention here? The concern about microplastics is not in human milk. It's not about preventing women from breastfeeding. It is about addressing the source of this particular microplastic and preventing them again from reaching mothers who need to breastfeed their infants. So it is important now to look at the source of the problem rather than just looking at the end result of a problem. So we need to be able to collaborate across different sectors. That is what is being proposed now by the United Nations Environmental Assembly, not only at the scientific level or the medical level, but to be able to do this at the community level as well and participate in prevention and solutions. So should you still allow should we allow women to continue breastfeeding when microplastics are actually present? I think it's very important to continue to breastfeed And of course, the precautionary principle tells us that when the weight of evidence tends to point to harm, then there must be some changes made in the way we recommend breastfeeding practices to our population. But it must be stressed, and this was actually emphasized by one of the authors who actually participated in the published study of October 2022 in Polymers. But it must be stressed that the advantages of breastfeeding are much greater than the disadvantages caused by the presence of polluting microplastics. Again, we also need to remember that there are sustainable development goals that may be invoked to reduce microplastic pollution, and these are covered under SDGs 3, 12, 6, 11, 13, 14, and 15. So we need to be able to repair ecosystems and retain biodiversity and reduce our plastic pollution footprints so that we can protect our present generation and the future generations. Thank you very, very much.
Our next speaker is Dr. Marisha Stella Recto. Dr. Recto graduated from UP College of Medicine and had her residency and fellowship training at the Philippine General Hospital. She underwent further postgraduate training in pediatric allergy and pulmonology at the University of Padova in Italy. She is currently a professor of the Division of Allergy and Immunology at UPPGH and is the section head of the Section of Allergy and Immunology at Asian Hospital and Medical Center. She is a fellow of the Philippine Pediatric Society and the Philippine Society of Allergy, Asthma, and Immunology. She's also a member of the European Academy of Allergy and Clinical Immunology, as well as the Asia-Pacific Academy of Allergy, Asthma, and Clinical Immunology. To discuss maternally transferred allergens in breast milk, can they protect or sensitize? Let us all welcome Dr. Marisha Recto.
Good day, everybody. My topic will be on maternally transferred allergens in breast milk. Can they protect or sensitize against allergies? These are my disclosures. Allergy conditions are affecting many people worldwide. In fact, 1.4 billion people already suffer from several forms of allergies. The allergy epidemic has started to become worse in the 21st century. In the 1990s, the first wave of allergies were on allergic rhinitis and asthma. But lately, in the year 2000, we see an increase in the skin allergies like atopic dermatitis and food allergies. And we see also here a parallel increase in the number of immune-related conditions called non-communicable diseases, of which allergies are one of them. So why is allergy such a big burden on society? Many children are already sensitized to allergens early in life, even without exposure. And it is highly associated with atopic comorbidities. It can present with severe life-threatening manifestations like anaphylaxis. Because of its chronicity, can have a significant socio-economic burden on society. And more importantly, it has no known cure because of its pathophysiology. So this is the atopic march, which is the natural progression of atopic conditions in a genetically predisposed individual. The earlier allergies are those of eczema and food allergy, which gradually get better with time but never totally disappear. And as the child gets older, you could see the emergence of respiratory allergies like asthma and allergic rhinitis, which may become lifelong conditions. Allergies develop because of an interaction of the genes with the environment. And we see here that there are several factors in the environment called the exposome, ranging from allergens to food or airborne allergens, from contaminants, pollutants, and even bacteria that will interact with the interfaces of our body to the environment, like the skin, the airways, or the gut mucosa. And this initial interaction will now dictate the emergence of what we call a Th2 rather cytokine profile that will allow a person to respond to the antigen either in an IgE mediated manner or into eosinophilia that will cause allergic disease. Now, epigenetics is very important to recognize in that it can shape the immune response of an otherwise non-atopic individual to become atopic. And this will pass on to the progeny and thus will cause more chronicity of disease as well as increase in the number of people who are having allergic conditions. Epigenetic modulation of gene expression occurs in both the pre and postnatal period. So here in this slide, you could see that even in utero, there is already effect or influence of the environment through the mother, either through her diet, through what microbes she's exposed to, and other pollutants that will influence the immune system of the child in utero. And at birth, there would be ongoing environmental modification, like changes in bacterial colonization the child will experience, or depending upon the diet that the child will have, like if the patient was breastfed or not, or the environment again, like pollution, which will affect the way his genes will be expressed. And we would now have the establishment of an allergic phenotype in that child, which may be passed on to his progeny and thus increasing the epidemic of allergy that we are now experiencing. So food allergy is one of the prototype allergies that are seen in children rather. And although it is known to be an IgE-mediated condition, we should think of it as a loss of tolerance to food antigens. And other allergies are also occurring because of a loss of tolerance. So food antigens normally in a healthy individual will be tolerated. There will be no immune reaction against them. And this is nature's way of allowing us to survive. However, in the atopic infant, you could see that the response to the food antigen will be dangerous in the form of an allergic inflammation. And this is because of their genetic predisposition. The two states of tolerance and inflammation actually are in a flux, meaning that patients with oral tolerance may eventually have a break in tolerance and make them allergic to the antigen, or those that were previously allergic to some antigen may develop tolerance and now tolerate the food. The principles of oral tolerance induction are very important to remember because this is going to be the basis by which we will justify the use of a maternal exposure to antigens on the child. So oral tolerance is an antigen-driven physiologic process that is resulting in an immune response to food antigens occurring in the gastrointestinal lymphoid tissue. And there is a very important critical window of development between 4 to 6 months of age wherein we can shape or modify the capacity for tolerance in these children by providing optimal colonization. There's also a genetic factor involved, the type of allergen exposed, the presence of early feeding, especially breastfeeding, and other immunomodulatory factors that can induce oral tolerance. So the process of oral tolerance is initiated by antigen exposure in the gut-associated lymphoid tissues. So this will lead to a regulated immune reaction. In this diagram, you could see that the antigen, which is always a protein, will now be accepted or will permeate through the epithelial barrier and will interact with the cells in the mucosa, wherein you could see that there would be normally either an immune response that will lead to deletion of immune responses or anergy, or it will lead to the development of tolerance, meaning that there will be T regulatory cells that are developed or produced in response to the antigen that will lead to a quiet immune reaction. However, allergies will occur in a person if there is a genetic predisposition that will enable an active immune response to develop. So this is what we want to achieve, that there would be no reaction to the antigen. So the gut-associated lymphoid tissue is a very important immune organ. It's the largest immune organ in our body, and it is the first interface that the child may have with a food antigen. So you could see that it is not just the immune cells that are important in the gut-associated lymphoid tissue. It is also the presence of the microbiome, which we should enhance in the child so that there would be an interaction between this in promoting oral tolerance. A very important concept to remember in the development of oral tolerance is the dual exposure hypothesis. And this means that it depends upon how the antigen is presented to the person so that an immune reaction will develop into tolerance or not. So it has been found that those children who are exposed to the antigens orally will allow the antigens to pass through the gut-associated lymphoid tissue. And as I mentioned earlier, the phenomenon of tolerance will develop. However, in atopic children, especially those with eczema or very severe skin conditions, the cutaneous exposure will not allow passage through the gut-associated lymphoid tissues, and thus tolerance will not occur. You would have activation of your Th2 cytokines and allergies will occur. That's why in the atopic individual, it is very important that we enhance oral exposure rather than cutaneous exposure. And this implies also that we have to take care of the skin barrier in children who are having severe eczema because they may develop food allergies later on. So infant feeding practices have indeed evolved through time for allergy prevention because of all this new data. Initially, we were always taught that allergy prevention must involve avoidance of food antigens. But now with the new concepts of oral tolerance, we can now see that there is really a promotion of oral tolerance that experts are recommending, and that delayed introduction of food antigens beyond 6 to 10 months will actually be detrimental towards the development of allergic disease, whether food allergies and other allergies later on in life. So these are different potential strategies through the diet in the primary and secondary prevention of atopic dermatitis and food allergies, because these are the first allergies that the child will experience. No? And you could see here breastfeeding tops the list. So breastfeeding, as you've heard in this convention, has many important roles, not only in nutrition, but even cognitive development. And now we will focus on the immune development of the child. So human milk is known to provide good nutrition and other psychologic benefits to the child. For the immunologist, human milk is actually an immunomodulating fluid that provides immune protection and regulation against different kinds of conditions. So here in this slide, we could see that the effector responses are present because of the bioactive responses against infections and also pathogens. However, there are certain antigens apparently in breast milk that may be sensitizing, leading to an allergic response. And it's not the food allergens, but the respiratory allergens like the dust mites. However, regulatory responses have been documented scientifically and through evidence that food antigens can actually cause immune activation in the physiologic sense and tolerance induction to allergens, and thus having no reaction, meaning that there would be no allergies to this if it passes through the breast milk. Breast milk actually contains many bioactive compounds that can instruct immune development. In this table, we see that there are inducing allergens like those I've mentioned, the airborne allergens like dust mite, and tolerizing antigens like food antigens. Also, the production of immune regulatory cytokines like TGF-beta and interleukin-10 are uniquely found in breast milk, as well as antibodies from the mother like secretory IgA and IgG, and chemokines and other cytokines, as well as vitamins and polyamines, all important for the induction of immune tolerance. This slide shows the unique antigens that are found in breast milk compared to formula-fed infants, showing that breast milk really is superior in the ability to provide tolerance to the individual or to the child. So here we see the different kinds of antigens, the variety of food antigens that the child can potentially be exposed to in breast milk versus the limited number depending upon what is being fed to the child. And also you could see the antigens in human milk are found in a variety of forms. Free forms, we see those that are in microvesicles and those that are bound to IgG and IgA in immune complexes that facilitate ready absorption through the gut-associated lymphoid tissues in comparison to that of formula. And of course, the immune modulators like bioactive molecules, which are not all found in formula, and also the variety of the microbiota, which the formula only tries to simulate, are really much different. So these are really pointing to the superiority of breast milk as an immunomodulating fluid compared to formula. So these are the different factors that can affect the immunologic shape of the infant. It can range from the antigens to the gut microbiome to the composition of the milk to the genetics of the person or the mother as well, and also to direct exposure to the infant's diet. So what we want to achieve is a balance, okay, an immune balance where intolerance would predominate over sensitization. These are the different factors in breast milk that can affect oral tolerance induction. Antigen shedding factors which can dictate the ability of the antigen to pass through the breast milk, like the rate of shedding, duration, maternal diet, the enteromammary reflexes or system, and the mammary gland permeability. The second would be the maternal immune status, wherein it all depends, of course, on the ability of the mother to have enough specific IgG and IgA, as well as immune complexes to facilitate the tolerance. And of course, the antigen dose, the nature of the antigen. As we've seen, there are those that which are tolerizing and those that are sensitizing, and repeatability of exposure, because you cannot develop an immune reaction without re-exposure. So after the antigens are exposed to through the mother, you could see different factors that can dictate this. And the antigen handling will also depend on the what we call the enteromammary system, wherein the mother will pass these factors from her enteric system to the child through breastfeeding, and of course, hopefully develop in the child oral tolerance. Let us discuss about some kinetics of food antigens in breast milk. Food allergens are detected in breast milk at very small amounts in free or immune complex forms. And this is important for oral tolerance induction. But even these small amounts in an already sensitized infant may cause allergic responses. So in children who are very allergic already, they have to be deprived of this antigen from the mother's milk. Antigen levels can also be seen as early as 10 minutes up to 4 hours after maternal ingestion. We have to remember, though, that there is a great variability in the presence or concentration of food antigens in breast milk from woman to woman. There are also factors that can influence kinetics of breast milk, like the type of food preparation, the mode of cooking, for example, in peanuts, whether it's roasted or boiled, the timing of ingestion, and the food matrix, whether the mother has started breastfeeding on an empty or a full stomach. And this can influence the way the breast milk is removed or released rather to the infant. The evidence that breast milk may be protecting or sensitizing is quite controversial and conflicting. There are evidences that there is an allergic protective effect of breastfeeding found in some meta-analysis against atopic dermatitis and cow's milk allergy. However, there are other long-term birth cohort studies that have demonstrated that breast milk may not protect against asthma or bronchial hyperresponsiveness in atopic individuals, or that it will not reduce the risk of allergic diseases in later life or sensitization, and may actually be detrimental in that it can increase the risk of sensitization and the emergence of allergic diseases. A meta-analysis on the role of breastfeeding in asthma and allergies was done in 2015, involving 89 studies. And the results show that there was low-grade quality evidence, especially its impact on medium to low-income countries, that the longer duration of breastfeeding was associated with a reduced risk of asthma in children aged 5 to 18 years old. They also found a low to very low quality evidence that breastfeeding is associated with a reduced risk of allergic rhinitis in children up to 5 years of age and eczema in children up to 2 years of age. And they found no association between breastfeeding and the development of food allergies. So the authors concluded that there was weak evidence that breastfeeding, regardless of length or exclusivity, is protective for allergic disease. They also showed that there is evidence of greater protection in middle to low income countries and no evidence that these associations were modified by an allergic family history. The protective effect for the different allergic conditions appeared to be greater in early life, raising the possibility of mediation through protection from viral disease, which is actually a known trigger for allergic conditions, or misdiagnosis of viral disease, or its effects were only on specific phenotypes. And they also concluded there was weak evidence for the waning protection or the increased risk of developing allergies in older children. The authors, though, pointed to limitations of their meta-analysis. And the most important is that the kind of studies, as I've mentioned earlier, were not very ideal. They had inherent biases because they were observational studies and difficulty in controlling their confounders. The studies were also heterogeneous. There was a lot of recall bias with some participants, like the mothers, having problems recalling from the time they breastfed until the emergence of allergic diseases. And of course, the phenomenon of reverse causation, wherein those that tended to have severe allergic conditions would breastfeed longer and thus can confound the results that will come out of the studies. So despite all these controversies and conflicting data, there were some recommendations of breastfeeding by different international groups. The first recommendation that I will be tackling is by the AAP in 2019. Their current data says that they do not think that there's a direct relationship of breastfeeding on food allergy because of insufficient data to make recommendations. Currently, there's also no evidence, though, to delay the introduction of allergenic foods, whether through breastfeeding or through oral feeding beyond the period of 4 to 6 months, which is the critical period for oral tolerance induction to prevent food allergies and other allergies in later life. So there should be no recommendations for restricting the diet during pregnancy or lactation to prevent allergies. However, if you choose to introduce food earlier, like in the 4 to 6 months period, continued breastfeeding must be done until the period of 2 years of age. And breastfeeding, despite all the controversies or inconclusive evidence, is still highly recommended because it prevents atopic diseases as well as numerous other health benefits for the infants and the mothers. And other international guidelines have followed suit. You could see here that in the Western world, maternal diets are not restricted, and in breastfeeding, for allergy prevention, they would really want still exclusive breastfeeding for at least 4 months and giving the food maybe at around 4 to 6 months. But some have shown that they are not recommending for or against breastfeeding because mainly of the lack of data or clear clinical evidence. In the Asia Pacific, we also have our own recommendations. In fact, the Philippines also has a recommendation, which is a consensus between PS Pagan and Philippine Society of Allergy, Asthma, and Immunology. We all recommend exclusive breastfeeding for the first 3 to 6 months of life, especially during the period of oral tolerance induction, because this is the period wherein we can do something to shape the immune response against allergies. We also have to consider that the guidelines especially in the Western world, may not apply to us in the Asia Pacific region. And that is because in the Western world, most of their problems are non communicable diseases. While here in our third world areas, we see that the infections and malnutrition are still mainly the bigger problem than the non communicable diseases. And thus, we should consider always the socioeconomic impact and effects on mortality and practicality regarding the recommendations for infant feeding in our situation now. So there are several known advantages of breastfeeding for allergy prevention. It has been known to reduce the exposure to allergenic dietary proteins. It can prevent infections that may initiate or exacerbate allergic disease. It can initiate activation of the infant's immune system, and it has an impact on development of the infant's microbiome. But there's still a lot of unknowns regarding the role of breastfeeding in allergy prevention, like the duration of exclusive breastfeeding and how it can protect against allergy, how the duration of any breastfeeding can impact on development of these atopic diseases, whether never versus ever breastfeeding will impact on the development of allergies, and whether the intensity of breastfeeding or bottle feeding can really impact atopic diseases. So we have to do really more research in this front. So in summary, allergies are chronic inflammatory conditions which are incurable and warrant preventive measures to improve quality of life and prevent disease sequelae. Breastfeeding is the most cost-effective allergy preventive strategy to develop oral tolerance to food antigens due to the presence of several bioactive immunomodulating compounds. The ability of antigens found in breast milk to produce oral tolerance are affected by maternal factors like immune status and genetics, antigen shedding mechanics, the innate nature of the antigen, and the normal microbiota of mother and child. It is important to know that variability exists from woman to woman in allergen and tolerogenic factor levels in their breast milk that can contribute to the controversy over the prevention of allergic disease via breastfeeding. And more importantly, maternal influence on neonatal tolerance induction through breastfeeding is probably of great importance because of dietary and environmental antigen transfer through breast milk, and that there are the pleiotropic effects of breastfeeding on the gut and immune system maturation. Thank you very much for your kind attention and have a great day.
For our third topic for this afternoon session, we will have a panel of speakers who will talk about the effects of breastfeeding on future eating behaviors, non-communicable diseases, and intelligence. To start off, our first speaker is a graduate of Doctor of Medicine at University of the Philippines College of Medicine. She had her residency training in pediatrics and fellowship training in pediatric gastroenterology and applied nutrition at Philippine General Hospital. She also had fellowship training in pediatric clinical nutrition at the Hospital for Sick Children in Toronto, Canada. She is currently the chair of Nutrition Advocacy Committee, Philippine Pediatric Society, Southern Tagalog Chapter, a practicing pediatrician at St. Dominic Medical Center, Our Lady of the Pilar Medical Center, and San Pedro Calungsod Medical Center, and the past president of the Philippine Society of Pediatric Gastroenterology, Hepatology, and Nutrition. Let us all give a warm round of virtual applause to Dr. Josie Ragacion.
The first smile, the first word, the first step. These are important developmental milestones in the life of a child. But can the first days outside the womb also confer significant milestone, particularly in the eating behavior of our children? A pleasant day to each one. I'm Dr. Josie M. Rogacion, and I'll be talking about the effects of breastfeeding on future eating behaviors as part of the evidence update in the program. I have no relevant conflicts related to this presentation. And my objectives are the following. First, I'll be talking about the determinants of eating behavior. the effects of breastfeeding on eating behaviors, particularly focusing on food preferences, the effect of maternal diet during lactation with regards to food preferences, and also the effect of duration of breastfeeding on food preferences. Next, I'll be talking about breastfeeding as a bridge to complementary feeding. And lastly, I'll be talking about the long term effects on eating behaviors of early breastfeeding. Eating behavior is a complex interplay of several factors: physiological, psychological, environmental, and genetic, all influencing the different behaviors related to eating, like meal timing, the quantity of food intake, food preference, and food selection. For the different factors affecting eating behaviors, physiological factor refers to the sensation of hunger and satiety. Psychological factors pertain to the learned food preference, the knowledge of eating, and as well as motivation to eat. Environmental factors focus on food availability, how food is provided, and even the plate or portion size. Whereas genetic refers to taste preferences, particularly for sweet or salty foods which are acquired early in life. In addition, eating behavior is also indirectly molded by observing the influences of others around the child. The family members' eating behaviors serve as a model for child's development and eating behavior. The role of breastfeeding in eating behavior, based on these different factors, is actually centered on the psychological aspect. And this pertains to the role of breastfeeding in learned food preferences. So I'll be talking about more of food preferences in the next few slides. Now, food and taste preferences develop early and form a continuum, starting in the prenatal period, where the fetus is exposed to different flavors and tastes passing through the amniotic fluid. And this is followed by early exposure through breastfeeding, where the diet of the mother places an important role in in flavor tasting, and moving on to the complementary feeding stage. In short, breastfeeding is at the crossroads between prenatal exposure and complementary feeding. So, breastfeeding is considered as a flavored bridge between prenatal and complementary feeding stages. Now, the fact that prenatal and breastfeeding stages occur early in life, these are mainly influenced by maternal diet. So maternal diet plays an important role in shaping food preferences prenatally and breastfeeding and during breastfeeding. Whereas for complementary feeding, maternal knowledge and food preferences, as well as parenting style, all play important role, not much regarding maternal diet. And what is the underlying principle behind flavor or food preferences in the early stages of life? And this is regarding repeated exposures, which I will tell you in the next few slides. So, repeated exposures is the key in all stages, from prenatal to breastfeeding and complementary feeding. Taste and flavor senses are well developed at birth. Even the olfactory and gustatory senses are already well established at birth. No? But did you know that an adult's tongue has about 2,000 to 4,000 taste buds, while babies have 10,000? So they taste better than adults do. And also, did you know that babies have taste buds in their cheeks? Because these 10,000 taste buds are well distributed around the mouth, at the back, at the palate, the back of the mouth, and even the inner side of the cheeks. Also, there is innate preferences for sweet taste and aversion for bitter taste. As shown in this landmark experiment, uh, pertaining to the different facial expressions when sweet taste is introduced as contrasted to bitter taste. Also, there are genetically determined individual differences in taste preferences. At the same time, aside from the different preferences in terms of sweet tastes, preference for high energy foods, particularly high fat diet, is common among children. It is also common among young children to reject new foods, to learn to associate food flavors with post ingestive consequences of eating. So, what do we mean by post ingestive consequences of eating? Let's say, for example, if the child ate mango, which is sour, so this elicits some negative effects or negative feeling in the stomach of these children. Naturally, he will try to avoid eating that particular food because it has generated some adverse events in the child. Now we all know, or we have been used to these different regions of taste in our tongue, no? But at the moment, this is not true anymore, because what is currently known is that all of the different tastes can be tasted everywhere in the tongue, and the most sensitive areas are in the sides of the tongue, except for the bitter taste, where most of the sensory receptors are also concentrated at the back of the tongue. So they are in similar places, but in terms of how they perceive the taste, that's where the difference lie. Now let's go to breastfeeding and its effects on eating behaviors. I have here three important concepts. Number one, early development of food preferences may be related to breastfeeding. Second, breastfeeding sets the stage for complementary feeding. And lastly, food preferences or behaviors developed by breastfed infants are carried over to later life stages. So let's talk about the first concept, and that is with regard the relationship between breastfeeding and development of food preferences. We know for a fact that breastfeeding not only contain nutrients in terms of macronutrients like carbohydrates, protein, fats, vitamins, and minerals, and also bioactive components, but more especially, it also contains flavors. And these flavors are coming from the food ingested by the mother, not only food, but also beverages, including alcohol. And not only ingestion, but through inhalation. The flavors are also the flavors also travel through inhalation and then goes to the breast milk in particular. Now, with regards early development of food preferences, those infants who are exposed to more varied flavors in breast milk are having this sensation because of the transfer of the different flavor compounds to the breast milk. And that breast milk has a lot of tastants or other volatiles from the mother's dietary and environmental exposures. But what is important is that repeated presentation of these flavors through breast milk influences the infant's feeding behaviors and preferences immediately after birth. Now, early flavor experiences are related to exposures to different milks. No? It's not just breast milk, but also even non-breast milk, but they may shape early food preferences. However, the most important concept that I'd like to impart is that there are differences on how the child prefer food, whether he will be breastfed or formula fed, because of these following characteristics. Now, in terms of flavor profile, breastfeeding is always dynamic. It's ever changing, and also the flavor varies within and between mothers. On the other hand, formula feeding will have constant flavor profile. It's rather monotonous since mothers only stick to one brand, no? In terms of the source of flavors, the source of flavors for breastfed infants is, of course, mother's diet. But for non-breastfed, it will depend on the type of formula. So it will be either cow's milk or non-cow's milk. Now, the good thing about breastfeeding or breast milk is that the predominant taste quality is more of sweetness. But for formulas, it will depend on the type of the milk, no? because cow's milk-based formula have low sweetness and sour taste. The soy-based basically have combination of sweet, sour, and bitter, whereas your extensively hydrolyzed are basically sour and bitter, such that if the child is introduced to this sour and bitter taste from this extensively hydrolyzed formula early on in life, they will preferably they will prefer sour and bitter taste also later in life. Now, the effects of food preferences are also affected by the maternal diet during lactation. In one of these experiments, wherein 75 lactating mothers were randomized into 5 groups, they were given vegetable juices, either broccoli or carrot juice, in 5 different ways. No, you have for the first group vegetable juice for 3 months, and this occurred between zero point 0.5 to 3.5 months postpartum. The other group received vegetable juice for 1 month, 0.5 month postpartum. The other one, the third group, received vegetable juice for 1 month, 1.5 months postpartum, and so on until until 2.5 months postpartum in group 4, whereas the control group only received water 0.5 to 3.5 months postpartum. So what was the findings from this experiment or from this study? And the infants were assessed at 6 to 8 months of age. What the authors found was that significant consumption of carrot-flavored cereals were noted among those infants whose mothers consumed the juice at zero point 0.5 to 1.5 months versus controls. There were no differences between controls and those exposed at 1.5, 2.5, or 2.5 and 3.5, meaning to say, beyond half month postpartum, there were no differences. There were also no differences in consumption duration, meaning to say, between 3 months and 1 month, and no effects on the amount of intake, meaning to say, there no effect on how much the child would take or the rate of feeding of the child. So this basically boils down to the finding that the timing of exposure is more impactful than duration, and that early exposure is the one that makes the difference. Now, the other studies on maternal diet and food preference show that previous exposure to certain foods during pregnancy and lactation can predict liking for certain food odors by the child at a later age. In the previous slide, the child was exposed to the food itself, but this time this study tried to see the effect of food odors on food preferences. So what they did, the question that they posted was that, can maternal dietary patterns during pregnancy and lactation predict the child's preference for pleasant or unpleasant odors of foods at 8 and 12 months? So they assessed the children at 8 months, and it was noted that there was greater consumption of green vegetables by pregnant and lactating mothers, and this significantly predicted the liking for green vegetables by infants, but not for pleasant odors. And this was not observed by the 12 months. So the pleasant odors are basically those with the flavors like odors like coming from strawberry, from peaches, and unpleasant odors are coming from green vegetables, cheese, and fish. So the authors concluded, and then they implied, that maternal diet during pregnancy and lactation may promote preference for foods that are not typically sweet, but with intrinsically unpleasant flavors. But however, this association fades once the infant exposures expand via solid feeding later in childhood. The other thing about food preference is that it may be affected also by breastfeeding duration. We talk about the effect of maternal diet, this time the effect of breastfeeding duration. There has been a lot of observational studies, and this shows that longer breastfeeding duration predicts higher dietary quality during later childhood. However, most of these observational studies suffer from many methodological issues, specifically regarding occurrences of several confounding variables. There are only a few studies which try to control for these several confounders. And they found out that longer breastfeeding duration predicted greater vegetable consumption at 18 to 24 months, and that they consume it at greater frequency and more variety in terms of fruit and vegetable consumption at two different points, at 2 years and going on until 12 years. And longer breastfeeding duration is also associated with higher fruit and vegetable intakes at 12 months and at 6 years of age. And long breastfeeding duration, if coupled with greater maternal vegetable intake, best predicted higher child vegetable intake at 6 years, but not at 12 months of age. So let's go to the next concept, and this is regarding breastfeeding effect as setting the stage for complementary feeding. Let's look at this bear stage. No, this bear stage is actually can be likened to breastfeeding. So it sets the stage, but this stage is just the foundation of something that is more with trimmings and with all those necessary decorations for a complete stage. So breastfeeding sets that stage. And it is as part of postnatal sensory experience, because breastfeeding as part of this sensory experience influences the acceptance of flavors in solid foods that may facilitate weaning or complementary feeding stage. So here, infants prefer flavors experienced during gestation and lactation. So this best prepares the child towards complementary feeding, which in fact is a different transition stage, coming from just one source of food, which is breast milk, liquid in consistency, and the child is now transitioning to the variety of foods, a variety of flavors, as well as different textures during complementary feeding. So this is now the stage for complementary feeding with all the drapes, the backdrop, etc. So complementary feeding, in this stage, the flavor learning continues. The infants discover sensory and nutritional properties of foods. But the important considerations here are variety and repeated exposures. Because with repeated exposures, it creates familiarity with the food, with the flavor, with the taste. And at the same time, with familiarity, it increases acceptance to that particular food. And food acceptance and breastfeeding shows that children who were previously breastfed tend to be less picky and are more willing to try new foods during childhood. At the same time, they found out that fruits and vegetables are always the more problematic foods now with regards introducing this to young children. So if they ate more if they were repeatedly exposed early through maternal diet, not only during pregnancy, but also during lactation. And it takes at least 8 repeated exposures or occasions during the complementary feeding stage before the child can accept this new fruit or new vegetable. Now, in this landmark study that also utilized the concept of continuum, no? Meaning to say exposure during prenatal can go a long way towards building preferences during breastfeeding stage and towards complementary feeding stage. So when they tried to give carrot juice to pregnant mothers and then went on to also continue giving the carrot juice to the breastfeeding child, what they found out that during the complementary feeding stage, it was not difficult to introduce the carrot juice among these children who were started on complementary feeding, such that prenatal and early postnatal exposure to flavor enhance the infant's enjoyment of that flavor in solid foods during complementary feeding. Now let's go to the last concept, and this is with regards the food preferences and behaviors being carried over to the later life stages. In a recent systematic review on breastfeeding duration and eating behaviors at 2 to 6 years old that was published just recently this year, 2023, uh, it noted that it involved 7 out of 26,000 articles that were published between 2011 to 2020. 4 were cohort and 3 were cross-sectional studies with a total of 13,000 subjects. However, due to some methodological issues, it was not, I think it was not possible to do a meta-analysis, no? Because there was lack of standardization to assess exposure and outcome. Meaning to say, exposure to breastfeeding, the definition was the duration of breastfeeding are different across all these studies, and outcome in terms of the questionnaires or the instruments that were used were also different. There were also differences in sample size and significant age range at outcome assessment. And for one retrospective study, of course, there is a possibility of memory bias. And at the same time, it will also be dependent on the parental perception of child's eating behavior. Now, despite those limitations, we can still say that the findings were still significant because out of the 7 studies, 4 out of them showed a positive effect of breastfeeding duration on child's eating behavior, such that these children who were previously breastfed had healthier eating behavior, they have lesser food neophobia, they have lower scores on food responsiveness subscales, and they were also lower in terms of their picky eating behavior. Now, another study that was published after that systematic review came out, and this was in 2021, which was also a longitudinal observational study consisting of 107 mother-child dyads, 98% of whom were breastfed and 46% were not breastfed. And they assessed eating behavior using Children's Eating Behavior Questionnaire at 3 to 5 years of age. Now, this questionnaire, this has been validated for use in several studies. This questionnaire was divided into two, no? Food approach scale. The food approach scale is actually linked to obesity, and the food avoidance scale, which is basically related to food selectivity. And this study showed that there was significant association between lower food approach scale and total breastfeeding duration of at least 6 months and exclusive breastfeeding for at least 3 months. So basically, these children have lower food approach scores. And then when they adjusted for social demographics and infant nutritional status and made their regression analysis, they found out that a 1-day increase in total breastfeeding or in exclusive breastfeeding, this was associated with a decrease, 0.04 decrease in food approach score. Let me remind you that the food approach score is linked to obesity. So that is why perhaps this eating behavior that is related to breastfeeding may also have an impact on obesity occurrence in the future. And I think this will be discussed by the other speaker. Now, the last one that I'd like to focus on is on the other benefit of breastfeeding and future eating behaviors. And this may be related to the parenting style because breastfeeding may promote maternal feeding styles that are less controlling and more responsive to infant cues and hunger satiety. That is why this will allow infants to develop a greater self-regulation of intake. So let me end this with these key messages. come to think of all the things that I have mentioned, there are basically three recurring themes: early exposure, repeated exposure, and tracking of behaviors. Early exposures means to say there is the early food and flavor exposure will determine future food preferences, and that the underlying mechanism or the underlying key to this is repeated exposure, because repeated flavor or food exposures may lead to conditioning, familiarity, and acceptance of novel foods. And breastfeeding thus sets the stage for this particular important feeding stage, and that is the complementary feeding stage. And lastly, with regards to tracking of behaviors, eating behaviors later in childhood are attributable to early experience through lactation. So these recurring themes of early exposure, repeated exposures, and tracking are all encapsulated within breastfeeding. Now, let me leave you with this quote. It is possible to educate children the pleasures of food, and that doing so will set the children up for a lifetime of healthy eating. And this can be achieved by breastfeeding. Thank you very much for your kind attention and have a good day.
Our next speaker is also a graduate of Doctor of Medicine from the University of the Philippines College of Medicine. She finished her pediatric residency training at Philippine General Hospital and had her fellowship training in pediatric endocrinology from the University of California, San Diego, USA. She also finished her master's in clinical medicine in child health at the University of the Philippines College of Medicine. She is an emeritus fellow of the Philippine Pediatric Society and the past president of the Philippine Society of Pediatric Metabolism and Endocrinology and Philippine Association of the Study of Overweight and Obesity. Currently, she is a consultant at various hospitals in Metro Manila, such as Cardinal Santos Medical Center, Manila Doctors Hospital, Chinese General Hospital, the Medical City, Our Lady of Lourdes Hospital, and Mandaluyong City Medical Center. Let us all welcome Dr. Shok Sowan Kua.
Good day. First of all, I'd like to thank the organizer for inviting me to be a speaker in this 10th Breastfeeding Congress. My topic is on the effect of breastfeeding on future non-communicable diseases. I'm Dr. Shoksan Chan Kua, a pediatric endocrinologist. These are the objectives. World Health Organization recommendation, breastfeeding for 6 months. Breastfeeding also contributes to a lifetime of good health. Adolescents and adults who were breastfed as baby are less likely to be overweight or obese. They are also less likely to have type 2 diabetes mellitus. What are non-communicable diseases? They include obesity-related conditions and cardiometabolic diseases such as hypertension, dyslipidemia, diabetes mellitus. They are the major causes of mortality in the world nowadays, notably in low and middle-income countries. The Philippines is a middle-low-income country. Are our Filipino children healthy? In the recent Pediatric Society Convention 2023, Dr. Mario Campansana of the Food and Nutrition Research Institute presented these slides. Overweight and obesity in school children was noted to be increased to 14% in 2021. Non-communicable diseases have a complex etiology resulting from the interaction of genetic factors, gender, age, ethnicity, and environmental factors. Non-communicable diseases in adulthood origin in early life. And we are seeing more and more obese children and adolescents in the last decade. They are at risk to develop non-communicable diseases at young age. We believe this is due to the lifestyle change. Particularly during pandemic, they are more sedentary and they have increased caloric intake, including much more simple carbohydrates, sugar added drinks, and this will also really contribute to the prevalence of overweight and obesity in our children and adolescents. So we should focus on primary prevention of non-communicable disease risk factor because the treatment of non-communicable diseases are much more expensive, and the prevention and early control of risk factor would be more efficient and cost-effective than the treatment. There are many biological and epidemiological studies on beneficial effect of breastfeeding during infancy on chronic diseases in adulthood. I will discuss on obesity, diabetes, hypercholesterolemia, and hypertension. So breastfeeding and protective effect against obesity. We know that breastfeeding, the breast milk has nutrient composition that provides a balanced mix of macronutrients, the protein, fats, and carbohydrates that will be promoting healthy growth and development of our children. And there's also hormonal regulation. Breast milk contains hormones that help regulate appetite and metabolism, specifically the leptin that exists in breast milk but not in artificial milk formula. This could change growth factor and prevent formation of adipocytes, and it regulates infants' appetite and satiety. Self-regulation. Breastfed baby have more control over their feeding. And better self-regulation of their hunger and fullness cues will lead to healthier eating habit later in life. In infants who were butterfilled, they tend to be overfeeding. Influence on food prevalences. The flavor of different food that a breastfeeding mother consumes can be transmitted to breast milk. Early exposure of infant to diverse flavor, and this will lead to infants' greater acceptance of a variety of healthy food during the weaning processes. WHO in 2013 had a systematic review on long-term effect of breastfeeding on non-communicable diseases. It was noted there was an association between breastfeeding and the lower prevalence of overweight and obesity later in life. The higher quality studies suggest a small reduction of about 10% in the prevalence of overweight and obesity in children exposed to longer duration of breastfeeding, particularly among the parents who are more educated with higher income. Their baby would be at a lesser risk to develop overweight and obesity. In this review published in 2014, was noted breastfeeding is associated with reduced risk of childhood obesity. But it also suggested that we need more attention in future birth cohort studies. A meta-analysis of 25 studies involving more than 200,000 participants in 12 countries. The results show that breastfeeding was associated with significantly reduced risk of obesity. And 7 studies reveal a dose-response effect between breastfeeding duration and reduced risk of childhood obesity. In this article, direct breastfeeding is inversely associated with weight gain velocity and body mass index or BMI. It was also noted the associations are dose dependent. However, the associations partially diminish when breast milk is fed from a bottle and substantially weakened by formula supplementation after neonatal period. In this study, overweight and obesity is less frequent among children who were exclusively breastfed for at least 6 months. They concluded exclusive breastfeeding time and maternal obesity have a significant effect on childhood overweight and obesity. Longer exclusive breastfeeding duration reduce the risk of childhood overweight and obesity, whereas maternal obesity increase the risk of their childhood overweight and obesity. This cross-sectional study involving more than 10,000 students aged 6 to 16 years done in Qingdao, China in 2017, the conclusion, breastfeeding can significantly reduce the prevalence of overweight and obesity among children and adolescents aged 6 to 16 years. Those who were breastfed for more than 12 months had the lower risk of developing overweight and obesity, especially boys between age 9 to 11 years. How about type 2 diabetes? World Health Organization in the systematic review showed that the evidence suggests that breastfeeding may have protective effect against type 2 diabetes mellitus, particularly among adolescents. However, it also notes that the study, small number, and there was also heterogeneity among study. In this review of 52 relevant studies, analysis of 21 articles on the topic, they involve the data synthesis, important risk factor for developing diabetes, duration and exclusivity of breastfeeding, early use of cow's milk, and also human milk contains substances that promote maturation of immune system, promotes satiety and energy balance, leading to preventing excess weight gain during childhood. So the conclusion of the review, the lack of breastfeeding can be a modifiable risk factor for both type 1 and type 2 diabetes mellitus. And the strategy aiming at the promotion and support of breastfeeding should be used by the trained healthcare professional in order to prevent the early onset of diabetes mellitus. Let's take a look at the cholesterol. Breastfeeding has been linked to improved lipid profile, an important marker of cardiovascular health. In this meta-analysis, a small beneficial effect of breastfeeding on total cholesterol in adulthood was noted, but it also suggests that future study to support a long-term programming effect of breastfeeding on blood lipid should be done. Let's go to the review published by this article in 2014. It was noted that breastfeeding has protective roles against non-communicable diseases, and the cholesterol from breastfeeding may have long-term effect on cholesterol endogenesis. It was noted to be related to the liver hydroxymethyl glutaric enzyme, or in short, HMG-CoA, which is a restrictive enzyme in cholesterol biosynthetic pathway. Its inhibitors have a blood cholesterol reducing effect. Breast milk is noted to contain hormones, particularly leptin, which we mentioned earlier, and that involved in the appetite regulation and energy balance in breastfed infants. How about blood pressure? This review show a small protective effect of breastfeeding against systolic blood pressure. But there's some confounding residual factor cannot be ruled out. Let's go back to this review published in 2014. The effect of breastfeeding on BP in adulthood can be partly explained through the following mechanism: reduced sodium intake in infancy, high content of long-chain unsaturated fatty acid in breast milk, protection against hyperinsulinemia in infancy, prevention of insulin resistance in early life, adolescence, and adulthood. Some conflicting result exists in this regard, so it needs longer study with long-term follow-up. Let's go to the last objective. So it's important to encourage and support breastfeeding. We should inform the parents about the benefit for babies and mothers. These are the benefits for baby and benefit for mother. So we should emphasize, encourage direct breastfeeding and aim for 6 months duration. Here, I would like also to emphasize avoid prolonged use of plastic bottle because of the endocrine disruptive chemical that can lead to not only pediatric obesity, but we also noted in our local setting, we've seen increased prevalence of precocious puberty, particularly among obese girls. When we say precocious puberty, that means the onset of secondary sexual characteristics in female is earlier than 8 years of age. In the recent years, we have seen more and more girls come in with precocious puberty, particularly those who have prolonged bottle feeding with the plastic bottles. In our local setting, I have patients who were bottle fed till 8 years of age. We would discourage really the prolonged use of plastic bottle in feeding. So in conclusion, breastfeeding is associated with better cardiometabolic health outcome, and it will have reduced prevalence of non-communicable diseases in children and adolescents, with reduced risk of obesity, decreased risk of developing diabetes mellitus, improved lipid profile, and lower blood pressure. Take-home message: we would encourage and support breastfeeding, attain healthy weight and body mass index in children, We need to monitor the growth, weight, and length, and plot the growth and body mass index data in the chart. We would ensure optimal health and reduce risk of non-communicable diseases by encouraging and support breastfeeding. In obese children and adolescents, healthcare professionals should identify early the cardiometabolic risk. Thank you.
Our next panelist is a graduate of Doctor of Medicine at the University of the Philippines College of Medicine. She had her pediatric residency training and her fellowship training in pediatric neurology at Philippine General Hospital. She also finished her fellowship training in clinical neurology and neurophysiology at Royal Alexandria Hospital for Children in Sydney, Australia. She is currently a professor and chair of the Department of Pediatrics at the University of the Philippines College of Medicine, Philippine General Hospital. She is also a consultant at the following hospitals: Capital Medical Center, Manila Doctors Hospital, and Metropolitan Medical Center. To discuss the effect of breastfeeding on future intelligence, let us welcome Dr. Marisa Lukban.
Good evening. It is my honor and privilege to be invited to be part of this panel to speak on the effects of breastfeeding on future intelligence. I'm Dr. Marisa Lukban, a child neurologist, and I have no conflicts of interest to disclose. The objective of my talk is to look at all the available studies that aim to establish a link between breastfeeding and performance in cognitive tests or tests of intelligence. I will be referencing from actual published researchers and reviews taken from researchers in this field. The cognitive development of an infant is a complex process, influenced by multiple genetic as well as environmental factors that interact with one another. As the saying goes, nature and nurture confer intelligence. Nutrition is another piece in the puzzle that confers a child's future intelligence. Formula milk differ from human milk in several nutritional components that may affect the biological basis of mental function, including hormones and growth factors. The question therefore is the association between breastfeeding and intelligence attributable largely to differences in maternal intellectual ability and quality of parenting rather than the nutritional benefit of breast milk on neurodevelopment? All through the years, the challenge for researchers is to prove that breast milk promotes higher intelligence, not confounded by genetic and environmental factors. What are the mechanisms underlying the association of breastfeeding and intelligence? Bernard et al. In 2017 observed among children aged 5 to 6 years that full-scale IQ was higher among those children who had been breastfed. Children who had high IQ scores were found to be fed with colostrum with high levels of arachidonic acid and 3 long-chain polyunsaturated fatty acid, compared to children whose colostrum had low levels of AA and 3-LC-PUFA, showing only intermediate IQ levels. Those children who were never breastfed presented the lowest mean IQ scores. Belfort in 2016 looked at the neurological outcomes of preterm infants at 7 years of age and found that the number of days on which the children received more than 50% of enteral intake as breast milk was associated with larger, deep nuclear gray matter and hippocampal volume. Owu in 2016 noticed that gray matter volume in both parietal lobes and left temporal lobes were greater among children who had been breastfed. On the other hand, white matter and cortical myelination helps provide the foundation for brain connectivity and supports the emergence of cognitive and behavioral functioning. Early trajectories of myelination are associated with good cognitive abilities and outcomes. Breastfed children showed higher activation in the right frontal and temporal lobes for perception tasks and higher activation in the left temporal lobe for language tasks. As brain activation is positively correlated with task performance, these findings reinforce the evidence that breastfeeding is associated with performance in intelligence tests. Thus, early life nutrition is an important and modifiable factor that can shape myelination and consequently cognitive outcomes. The comparison of breast versus formula feeding is complicated because there are differences in first what is fed, breast milk versus formula milk fortified with DHA and AA. Second, who feeds? What is the contribution of maternal factors and rearing practices? And third, how is it fed? Breastfeeding versus express breast milk, is there a difference? As well as the duration and manner of breastfeeding. In other words, compliance or non-compliance to WHO recommendation of exclusive breastfeeding of 6 months. For example, mothers who succeed at breastfeeding are different from those who feed formula milk, and the process of breastfeeding itself has profound effect on the behavior and physiology of the mother and of the mother-infant dyad. Studies show that in general, breastfeeding mothers have higher socioeconomic status, better educational level, and higher intelligence. They provide an enhanced home environment and have more positive health behaviors. They have fewer symptoms indicative of depression and greater preoccupation with the infant development. Most studies on breastfeeding are observational cohort surveys or case control studies. Early studies use retrospective collection of data, which may result to recall bias. The challenge, therefore, is to account for the contribution of confounding variables to the outcome of intelligence tests. Now, let us look at all the studies that span from 1929 till 2019. The early studies by Hofer and Hardy in 1929, tests done at 7 to 13 years of age, grouped according to the length of exclusive breastfeeding, show that infants who were breastfed between 4 to 9 months were superior mentally and physically. Douglas in 1958 showed that infants with longer length of breastfeeding walked earlier or showed an inverse relationship of breastfeeding with achievement of motor milestones. Broad in 1972 and 1975 showed that breastfed babies have fewer speech difficulties and better reading ability at age 5. Lastly, children aged 5 to 15 years with learning disorders are less likely to be breastfed, according to a Menke study in 1977. The large cohort studies started to control confounding variables in their study designs. There are 3 retrospective large cohort studies that showed significant positive correlation with IQ tests. The UK MRC and SHD enrolled babies born 1 week in March of 1946, with 1,133 babies entirely bottle-fed and 1,291 babies never bottle-fed. Those who were never bottle-fed or were breastfed had better performance in the picture intelligence test at 8 years of age and higher scores in non-verbal ability, mathematics, and sentence completion at age 15 years. The second study, the UK CHES, recruited babies born 1 week in April of 1970, with babies breastfed for at least 3 months and showed positive correlation in the English picture vocabulary test at age 5 and higher scores in the British Ability Scale at age 10. Contrary to the UK study, the research by Ferguson in 1982, the New Zealand Dunedin Multidisciplinary Child Development Study controlled maternal IQ as a confounder. At age 3 years, there was no significant difference in age of reaching milestones for motor ability, verbal comprehension or expression, intelligence, and incidence of behavioral problems. However, at age 5 and 7 years, there was a small but significant effect on the Stanford Binet and Whisk-R for breastfed babies with a mean difference of 1.89 points. The next study, the New Zealand Christchurch Child Developmental Study, was the first study where the data were collected prospectively. The duration of breastfeeding was a significant predictor of later cognitive or educational outcomes. Breastfed babies for 8 months or more had 0.1 and 0.3 standard deviation higher scores compared to those never breastfed. As breastfeeding mothers are more likely to provide a cognitively stimulating environment for their infants, it has been suggested that breastfeeding could be a marker of parenting practices that promote child development, such that the positive effect of breastfeeding on cognition could be due to the family environment and not due to nutrition. In this meta-analysis by Horta, initially including 14 studies in 2013 and updated to 17 studies in 2015, they excluded those studies that did not adjust their estimates for variables measuring home stimulation, reducing the likelihood that the association of breastfeeding with intelligence was due to parenting and home stimulation. The forest graph analysis shown here showed that the pooled effect estimate showed a mean difference of 3.44 points higher scores in intelligence tests for breastfed children. Horta also showed that when the maternal IQ was controlled, there remains a small but definite benefit of breastfeeding with mean difference of 1.92 points in favor of breastfeeding. The study also showed smaller but significant benefit from breastfeeding for babies evaluated between 10 to 19 years of age. That's 1.92 mean difference compared to younger subjects less than 19 years of age with a mean difference of 4.12 points. The inclusion of 17 studies showed no publication bias, as the mean effects are similar for cohorts that have less than 500 subjects compared with those equal and more than 500 subjects. More recently, Horta looked for new studies that will reinforce the positive long effect of breastfeeding, specifically excluding studies that fail to adjust the estimates for socioeconomic variables and stimulation at home. In this 2018 publication, he identified two studies, those done by Bernard and Boucher, that reported the duration of breastfeeding was positively associated with IQ at 5 to 6 years of age. The study of Rantalainen reported that cognitive scores at 67.9 years of age was higher among those subjects who had been breastfed for at least 6 months. Furthermore, two studies reported that the small gain in IQ was associated with higher school achievement and income in adulthood. There are ethical issues in conducting randomized controlled trials on breastfeeding, if it is the subject or the babies who are randomized. There is a way to go around this, and this RCT was designed in such a way that the intervention, a WHO initiative on breastfeeding promotion, was made randomly to participating hospitals and polyclinics in Belarus. Those who were exposed to the intervention group led to a large increase in exclusive breastfeeding for 3 months, 43.3% for the experimental group versus only 6.4% for the control group. The breastfeeding promotion also led to a significantly higher prevalence of breastfeeding at all ages up to 12 months of age. The results showed that when the children were evaluated using a standardized IQ test, the verbal IQ, performance IQ, and full-scale IQ scales were positively higher among the breastfed children. The teachers' academic ratings were also significantly higher in those who had the breastfeeding intervention. But what do several points on IQ scores actually mean? Do these scores translate to educational and financial success in later life? A prospective population-based birth cohort study of neonates born in 1982 in Pelotas, Brazil, tried to answer this question. Victoria et al. Tried to evaluate whether breastfeeding duration has an effect on adult IQ scores at age 30, their educational attainment, as well as their income. The cohort was analyzed into three groups according to tertiles of family income at birth. Figure 1 shows a linear correlation with better IQ test performance as breastfeeding duration increases. The three separate lines representing family income at birth, green for low income, blue for middle income, and red for high income, support a strong association between IQ and breastfeeding duration, regardless of the family income at birth. Among the 3,493 participants for which data were available, the duration of breastfeeding was positively associated with higher IQ, educational attainment, and income. Look at the graph, and this data shows that there is a dose-dependent response on duration of breastfeeding, with higher IQ scores, more years of schooling, and monthly income for those who were breastfed for at least 6 months. Data from the British Avon Longitudinal Study of Parents and Children cohort study were used to assess the economic impact of breastfeeding. There was a positive association between breastfeeding and achieving higher educational attainment at 16 years of age, defined as having 5 general certificate of secondary education with grades of C or higher. Health economic models estimated that the average lifetime income was expected to be 67,535 higher for children achieving a +5 passing GCSE grades compared to those achieving less than 5 passing grades. Incorporating the impact of breastfeeding on educational attainment translates into an expected lifetime income of £4,208 higher for children who were breastfed for less than 6 months and £8,799 higher for children who were breastfed for 6 months compared with children who were never breastfed. Lastly, the latest systematic review on this topic was done by Mike Gowen and just published early this year. They reviewed articles from 2012 to 2019 and selected only those studies that adjusted for two important confounding variables, maternal IQ and home environment. 21 of the articles assessed the effects of breastfeeding on cognition, 5 of which adjusted for both maternal IQ and home environment, 9 for maternal IQ only, and 7 for home environment only. Of the 21 studies, 17 demonstrated a modest increase in cognitive scores in children who were breastfed. 2 studies demonstrated a positive dose-dependent effect between breastfeeding and the MSCA test scores of between 2.9 and 7.3, depending on the length of breastfeeding. Studies comparing exclusive breastfeeding and partial breastfeeding demonstrated a dose effect with exclusively breastfed infants showing higher cognitive scores per month of exclusive breastfeeding compared with infants who were only partially breastfed. The test score difference ranged from 0.19 to 0.96 points per month of breastfeeding, comparing any breastfeeding, predominant breastfeeding, and exclusive breastfeeding. In conclusion, to the question, does breastfeeding alter early brain development and future intelligence? I have presented the overwhelming evidence on the effects of breastfeeding to future intelligence. Newer studies are exploring the implication of higher IQ scores at age 5 to 6 years old to educational attainment and income at adult age. This evidence can be used to support public policies to increase breastfeeding in our country for the promotion, protection, and support of breastfeeding. Thank you for your kind attention.
Thank you very much to our speakers for that insightful lecture. We would like to welcome back Dr. Maria Isabel Quilendrino, together with our esteemed speakers, to the virtual floor for our question and answer forum. Thank you very much to our distinguished speakers. We would like to welcome back Dr. Sherry Suplido, together with our esteemed speakers, to the virtual floor for our question and answer forum.
Good evening. The important points raised by our speakers highlight the far-reaching, if not lifelong, implications of environmental factors and early nutrition on future health and developmental outcomes. Our speakers have presented current evidence for the superiority of breastfeeding in health and developmental outcomes. So to answer our questions live, we would like to welcome back our esteemed speakers, Dr. Recto, Dr. Ragacion, Dr. Kua, Dr. Lukban, and unfortunately, Dr. Macalinaw is not with us during this live Q&A, but we'll be collating the questions and we'll be attempting to have our speakers answer them after this forum. So for the first question, it is addressed to Dr. Recto. So you presented that new evidence no longer support dietary restrictions in mothers, in lactating mothers. To clarify, is there still a place for elimination or hypoallergenic diet in mothers whose infants have allergies, especially if severe, in a wheezing infant, for example?
Okay, thank you for that question, Isa. So it is very important that mothers eat a diverse diet while lactating. And this is because this will be the source of the antigens that will induce oral tolerance in the child. So you can just imagine a picky eater mother, what kind of child they will have, probably a very allergic child with a lot of food allergies. So this dietary introduction is very important early in life, especially in the first 4 to 6 months of life. However, this is more for the population that does not have, or the children that do not yet have allergies. Meaning, let's say they have already an established food allergy because they had eczema, and that through the skin, they were already sensitized. They have peanut allergies or they have milk allergies. And the mother will, of course, because of her diet, pass the antigen to the already sensitized infant. This can cause an allergic reaction. So only in these cases wherein there is a documented allergy already in the child to a certain food, which can only be found out through allergy testing, not through, surmising or maybe like giving some educated guesses. No? So we have to test through allergy testing if the child is really sensitized. And if it is so, then the mother is given a directed elimination diet. It's not a diet encompassing many allergens, but simply directed to the particular allergen, whether it be milk, protein, or eggs. But eventually, this is not a lifelong diet. As the child gets older, tolerance will develop. And that's why the mother would eventually, and if chooses to breastfeed, might already be able to take back the food and give it to the child later on. So as allergists, we monitor the emergence of tolerance. Now, tolerance will come out in patients who already have allergies if they have been taken away from the allergen for a while. And once we have some markers by which we can reintroduce, this will not last forever, except for peanut allergies. So there are two scenarios. One is the not sensitized, meaning the mother should eat everything so that the child will become tolerant. And those that are already allergic, they have to avoid it for a certain period of time, but eventually they will be able to resume eating.
Thank you, Dr. Rector, for clarifying that. So this is actually a recommendation that has to be made only after very careful evaluation and monitoring. So the next question is for Dr. Lukban. There is a trend for higher IQs in exclusively breastfed infants. In studies looking at young school-age children, were they able to establish an association with the duration of breastfeeding and this increased IQ scores?
Yes, I think in one of the slides, if you remember, I showed you a graph wherein there is a study that mentioned that the longer the duration of breastfeeding, the higher the IQ scores was. So the answer is yes. The duration of breastfeeding impacts into higher IQ scores when tested at in the school age group.
Thank you, Dr. Lukban. We'll move on to the next question in the interest of time. So for Dr. Kua, is there an impact of maternal obesity on breast milk quality? You mentioned in one of the studies that maternal obesity was associated with higher risk for child obesity. Is this finding true for breastfed infants as well?
Yes, maternal obesity can potentially impact on breast milk quality. There's some research suggests that maternal obesity may lead to changes in the composition of the breast milk, including the alteration of in the level of various nutrients, hormone, and immune factors. But we still need to know more study needed to fully understand the extent and specific mechanism of this effect. And then the other question is that maternal obesity associated with higher risk of obesity insurance. We know that if mama is obese, special, and having the children may be also higher risk because of the son genetic factor. But if you are breastfeeding your baby, we know in general, unlike you are feeding with the milk formula butter, they're really, you see much higher obesity can occur. So we also depends, also consider other factor. Let's say, would they be overfeeding, or when do they introduce a solid food, and also the family lifestyle and genetic and also hormonal factor have to be considered in the childhood obesity.
Thank you, Dr. Kua. So it's really interesting to see how many factors interact with each other in mediating the protective effect of breastfeeding and breast milk. So for our last question, addressed to Dr. Ragacion, can a mother compensate during lactation if she did not eat very well and only found out about the protective effect of dietary diversity during lactation and improving the food tolerance and preferences of her child?
Thanks, Isa, for that question. No? As I mentioned in the lecture, early flavor experience is very, very important, no, to develop future food preferences. No, ideally, it really should start prenatally. No, I have mentioned that over and over again that the diet of the pregnant mother is very important in the initiation of the flavor experience. Nonetheless, no, if the mother is not, getting a healthy diet during pregnancy, again, the next step is to do it during breastfeeding. Because I think those early exposures not only imply just prenatal exposure, but also the early feeding experience that is being experienced during the breastfeeding stage. So perhaps, no, I don't have the study. No, I don't have the study that, if you skip a healthy diet during pregnancy and compensate during lactation, it will eventually redound to, let's say, a better food preference in the infant. No, but personally, I think it's not too late. No, it's not too late if the mother realizes that she can do it during the breastfeeding stage, and really during lactation, what is also important, yung pa rin, no, Isa, yung transfer of the flavors, transfer of the odors. It's not just the flavor from the food itself, but also the odors during lactation, which is also important. So I think it's not too late for the mothers to, switch to a healthier diet during breastfeed while she's lactating, or during breastfeeding, to really initiate the early flavor experience in early infancy. So
Thank you,
pwede pa
Doc.
yun, Isa.
Yeah, so the earlier the better.
Yes,
And
the earlier, it's not just early, no, it's also variety. So
yes, and right.
repeated, and variety. Yun ang pinaka-importanting concepts in terms of flavor development or taste or food preferences.
Yes, and just as with any intervention, ever is never than never. So
Oh, that's right.
yes, thank you very
And,
much. Yes, but doctor.
no, it's okay. I'll
Okay.
just give the message later. Okay.
All right. Thank you very much to our esteemed speakers. Now we would like to award this certificate of appreciation, which reads, the Philippine Pediatric Society and the Philippine Obstetrical and Gynecological Society, together with the University of the Philippines, Philippine General Hospital, award this certificate of appreciation to Dr. Irma B. Macalinaw, Dr. Marisha Stella T. Recto, Dr. Josie M. Ragacion, Dr. Shoksoan C. Kua, and Dr. Marisa B. Lukban for sharing their expertise as speaker for the 10th Philippine Breastfeeding Congress entitled Roadmap to Breastfeeding Success, Creating Systems for Lactation Support, held on August 10 to 11, 2023. This certificate is signed by Dr. Aurora Gloria Libadia, overall chair of the Breastfeeding Congress, Dr. Stella Marie Jose, co-chair of the Best Breastfeeding Congress, Dr. Resty Bautista, chair of the Scientific Committee, Dr. Marisa Lukban, president of the Child Foundation of the Department of Pediatrics of UPPGH, Dr. Lourdes Capito, president of the Foundation of Reproductive Care, Dr. Teresa Ribaño, chair of the Breastfeeding Committee of the Philippine Pediatric Society, Dr. Florentina T., president of the Philippine Pediatric Society, and Dr. Efren Domingo, president of the Philippine Obstetrical and Gynecological Society. Thank you. I would also like to thank the organizing committee of the Breastfeeding Congress for awarding me the Certificate of Appreciation. It is my pleasure to moderate and be part of this program. We thank everyone for listening and actively participating in this day's session. For some reminders, we would like to remind everyone to answer the post-test and evaluation forms. You can access them by clicking on the tab, and it will be open at the end of the sessions. For those paying delegates who are listening through YouTube channel, please answer post-test and evaluation forms to get your certificates. Link to the post-test and evaluation forms will be sent to you via email. We will now close the link to the breastfeeding handbook, and we will send individually to the emails of paying delegates the revised version with better quality photos. Thank you for your understanding. For the closing remarks of this simultaneous session, we would like to introduce and call on the Chair of the Department of Obstetrics and Gynecology of the Philippine General Hospital, Dr. Antonia E. Habana.
My dear colleagues, nurses, students, and allied healthcare professionals, good afternoon. I'm Dr. Maria Antonia Habana, Professor and Chair of the Department of Obstetrics and Gynecology at the Philippine General Hospital, University of the Philippines, Manila. In partnership with the Department of Pediatrics, it has been an immense privilege to host the 10th Annual Breastfeeding Congress. We extend our gratitude to the following: the Breastfeeding Committee of both the UP-PGH Department of OBGYN and the Department of Pediatrics, led by Dr. Stella Marie Jose and Dr. Aurora Gloria Libadia. The Philippine Pediatric Society, led by their president, Dr. Florentina T., and the Philippine Obstetrical and Gynecological Society, led by President Efren Domingo, who have all worked so tirelessly over the past year to organize this event. To Docrity for hosting the online lectures, and to our excellent moderators and speakers for sharing their time and expertise. And finally, to our audience, we sincerely appreciate your participation. Maraming salamat. For the past 2 days, we have gained a great amount of knowledge on the importance of breastfeeding and its impact on the mother-baby dyad, as they are at the core of our practice as obstetricians, pediatricians, and healthcare providers. We have integrated the roadmap to strengthen the legislation, monitoring, and enforcement of the national breastfeeding laws enacted in our country. As we reflect on how our institution has come over the years, and especially during the COVID-19 pandemic, I am confident that together we are able to strive towards better education, innovation, and training to both healthcare providers and lay people alike. On a personal note, I breastfed all my 3 children and can attest not only to its health benefits, but to the special bond that it builds. Congratulations again to everyone. Happy learning.
And this officially ends the 10th Breastfeeding Congress, reminding everyone that the post-test and evaluation form are now accessible. Good evening and see you again next year.